Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Rare Disease Disorders and CNS Drug Development, Paving the Way for Precision Medicine. My name is Vicky, and I will be your X-Talks host for today. Today's webinar will run for approximately 60 minutes with time for a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. And at this time, all participants are in a listen-only mode. Please note that this event will be recorded and made available for future download. And at this point, I would like to thank Medpace, who helped develop the content for this presentation. Medpace is a scientifically driven global full-service clinical contract research organization providing phase 1 to 4 clinical development services to the biotechnology, pharmaceutical, and medical device industries. Medpace's mission is to accelerate the global development of safe and effective medical therapeutics through its physician-led, high science, and disciplined operating approach that leverages regulatory and deep expertise across all major therapeutic areas. The company has specialized expertise in clinical development for CNS as well as rare diseases. And now I would like to introduce our speakers for today's event. Our first speaker is Dr. James Vornov. Dr. Vornov is an MD, PhD neurologist with broad experience in both neuroscience and CNS drug development. He has particular expertise in the rapid transition of compounds from the laboratory to clinical proof of concept through the use of technologies such as biomarkers, PK/PD modeling, adaptive design, and clinical trial simulation. Dr. Vornov trained in neurology at the Johns Hopkins Medical School, where he served on the faculty for 10 years prior to transitioning to the industry. And our next speaker for today is Michelle Peterson. Michelle Peterson has 9 years of experience in clinical research, which includes managing global studies in phases 1 through 3 of development. She has managed multiple global rare and ultra-rare disease trials with a focus on neuromuscular and pediatric disorders. Ms. Peterson is a member of Medpace's Specialized Rare Disease Consortium and Patient Recruitment Team and holds a Master's of Science degree in Physiology. And our last speaker for today is Dr. Richard Shire. Dr. Shire is a board-certified neurologist and clinical pharmacologist with 20 years dedicated to clinical drug development. He has led clinical biomarker, companion diagnostic, and pharmacogenomic activities across therapeutic areas in large pharma and biotech. His experience includes macromolecular and genetic therapies for rare neurologic diseases. He received his medical degree from SUNY Upstate Medical University. And now, without further ado, I would like to hand the mic over to our speakers. They may begin when ready.
Well, I'd like to thank everyone for joining us for the webinar today. So the Medpace CNS group, I think, has had an interesting view of the changes in CNS drug development over the last few years. And what we'd like to do over the next hour is share that experience with you. We'll talk about how the understanding of the heritability of neurological diseases and psychiatric diseases is changing the landscape, and then talk a little bit about successes and failures in orphan and rare disease trials. Then we'll go on to the operational issues that we've experienced involving doing these types of trials, and then the current and new approaches that we're seeing in the development of targeted CNS therapies. Next slide. So in CNS clinical practice, we've always known that there's a lot of familial inheritance of CNS diseases. And when twin cases have been done, the cohorts generally confirm that there is a large amount of heritability of common neurologic diseases. Familial cases of CNS diseases have generally been thought of as being relatively rare and somewhat different than the sporadic cases that we usually see, but they've been studied intensely for hope that they could yield some insight into the pathology of the more common sporadic diseases. But in CNS disease, we see a lot of rare diseases. One of the first things that they teach you in medical school is that when you hear hoofbeats, you ought to think of horses and not zebras, meaning that a patient in front of you is more likely to have a common disease than a rare disease. But in the neurology clinic, we see a large number of rare disease, and many of them are genetic in nature. Next slide. One of the interesting things, though, is that as we've understood the genetic underpinnings of these genetic diseases, we haven't always gotten the kind of insight into pathophysiology that we might like. This chart shows some of the diseases that we've worked on recently or have been in development. And at the top, you see some of the diseases which are single gene mutations, in which once the single gene mutation was discovered, the etiology of the disease is quite clear. Wilson's disease was known to be a disease of copper metabolism, and the gene codes for a copper transport protein. Similarly, in Duchenne's, the gene is a gene expressed in muscle, but it's called dystrophin because we don't really understand exactly how these mutations in dystrophin cause the degeneration and the distribution of weakness in Duchenne's. Then we have other diseases like spinal muscular atrophy, where multiple genes have been involved. They all lead to motor neuron loss, but the exact way in which they lead to cellular death selectively in motor neurons isn't really understood. Then we move down to the bottom. There are deletions like those seen in the developmental disorder Smith-Magenis syndrome, in which the actual location and identity of the gene in that deletion is not really understood, or how it leads to the disorders. And of course, the bottom two are two CAG repeat diseases, Huntington's disease, which was one of the first neurodegenerative diseases which we understood the genetic underpinnings, but it's now been several decades since that's been described, and we really don't understand fully how these CAG repeats lead to the neurodegeneration that occurs in adult life. Similarly, spinocerebellar ataxia, multiple genes are described. They lead to different syndromes, but all with the same neurologic deficits caused by the same kind of CAG repeat. Next slide. So I think that one of the best illustrations of where things tend to stand today as we look at more common diseases is Alzheimer's disease. Alzheimer's disease is generally diagnosis has been made clinically, generally not till late stages. And you can't make a clinical diagnosis in the prodromal early stages or in what trialists now call the preclinical stage, the stage of Alzheimer's disease before any clinical signs or symptoms are apparent. But because we have biomarkers, both CSF and PET, we've started redefining the broad category of Alzheimer's disease really to be amyloid disease, patients who are accumulating amyloid in their brains. And as we've now delved into being able to find the pathology, early on, we find risk factors like ApoE4, APP, and presenilin, which are in the amyloid pathway. And these allow us to identify patients, individuals who have no sign of disease, but are very likely to, when scanned or looked at for biomarkers, have the beginnings of amyloid disease, thus letting them be treated beforehand with primary prevention of amyloid accumulation. Next slide. So this sort of summarizes how we approach common diseases as we understand their underpinnings. We start with the syndrome, the behavior, signs, and symptoms, where it's localized in the brain, and then go to the functional and anatomic substrate of the disease. What parts of the brain, what types of cells are affected, and what's the actual pathology causing the disease, and then down to the level of pathogenesis. what are the metabolic pathways, what's causing cell death or dysfunction, and then the genetic risk factors, environmental risk factors that lead a patient in neurodegenerative diseases, generally late in life, to manifest the disease. But to treat these kind of complex, multifactorial diseases, what we seek is a final common pathway, trying to either reverse the pathology that's occurring in the brain, or in something like primary prevention, prevent the pathology from expressing itself, or for most of the treatments that we have for neurologic diseases, actually just mitigate the symptoms by improving the function of the systems that have been affected by the disease. Next slide. So in the last couple of years, our understanding of the genetic underpinnings of neurodegenerative diseases has really changed. And I think this is part of the change in how we see the landscape and how we're now starting to approach doing clinical trials in these diseases. So ALS and frontotemporal dementia were thought of as two different diseases, but actually if you look back in the literature, all the way to the days of Charcot, it was realized that some patients with ALS had additional features, particularly cognitive decline. A gene, TDP43, a highly conserved gene with DNA, RNA, protein housekeeping function, was associated first with tau pathology in some FTD patients, but then as the gene became known and it was looked at in ALS patients, it was actually found in some familial ALS. We now understand that there's basically TDP43 diseases, a spectrum from ALS to frontotemporal dementia, and spectrum in between that may include both. There's the C9orf72 triplet repeat, which similarly can cause either ALS or can cause FTD or combinations of both. So of course, ALS has its own collection of mutations that have been discovered, SOD being perhaps the first in a familial ALS cohort causing ALS. So we end up with a picture where there are many genes that can produce either in familial or in sporadic disease, all leading to the same syndrome or set of syndromes. Next slide. Parkinson's disease similarly has seen an absolute explosion of the number of genes implicated, first from familial Parkinson's disease, and then quite fruitful studies of genome-wide sequencing or exome sequencing in sporadic patient populations. And it's interesting that it's a collection of variant risk factors, but all of these risk factors fall into categories in which investigators, especially creating animal models of Parkinson's disease, had been investigating for some time. We've known that there's a synuclein buildup, so there are abnormalities in some patients in the alpha-synuclein gene. Perhaps a surprise that came out of epidemiologic work was that the gene that's responsible for Gaucher's disease is also mutated in some patients with Parkinson's disease. The connection was made because patients with Gaucher's families were found to have a higher incidence than expected of Parkinson's disease. And then, of course, there are the PARC genes, which are linked to mitochondrial function. So again, probably multiple genes, multiple pathways, and multiple ways to cause the same clinical syndrome of Parkinson's disease. And of course, this pathology of Lewy body dementia, classic in Parkinson's disease, also can just cause Lewy body accumulation, can also cause dementia. So we come in something like Parkinson's disease thinking about, are these mutations risk factors that all lead to the same kind of process? Or are they actually signs that there's a genetic defect that with aging leads to a actual genetic disease of late life? Next slide. So these kind of findings are making us rethink our broad view of how we ought to approach these diseases as we understand genetic underpinnings. Perhaps they really are not single syndromes that ought to be approached by a common final pathway, but actually treated as separate diseases with different approaches depending on what's causing the disease in an individual patient. Next slide. And this has been called the precision medicine approach, where we start off clearly with the syndrome, localization, behavior, signs, and symptoms, but then we subgroup that patients based on their genetic diagnosis, what metabolic pathways are involved, what cells are being killed or not functioning well. And of course, in some patient groups, there may be other genetic factors that are either worsening, speeding the disease, or actually maybe protecting some patients from the disease. And then, of course, there's still things like aging, environmental risk factors probably are interacting with the genetic background. And then based on the subgroup, we would then choose a targeted therapy that would prevent that particular pathology or reverse it or mitigate the pathology specifically based on what pathology was driving the disease in a particular patient population. So next slide. Of course, in rare and orphan diseases, this has become a familiar approach because those diseases generally start with understanding that there's a genetic mutation driving the disease and then an obvious target to treat that disease. And rare diseases have been driven in development in part by the regulatory environment, which has encouraged the approval of the development of these very small patient populations. Next slide. We've seen a growth over the years in orphan drug designations and a steady stream of approvals of orphan drugs. Next slide. If you look at where orphan drugs are being, or rare disease drugs are being developed, oncology has become a major area, mostly because these are somatic mutations that are identified and then targeted. And of course, the other large category is classed as genetic disorders, but of course that includes a lot of genetic disorders with CNS manifestations. Next slide. So there have been great successes in orphan and rare diseases, and actually 41% of the 2016 FDA drug approvals were to treat rare diseases. And one of the earliest and greatest successes in treating rare diseases has been enzyme replacement therapy for inborn errors of metabolism due to genetic defects. Cerezyme was approved in 1994 for Gaucher's disease, but type 1, non-neuropathic disease. Elaprase is approved in Hunter syndrome, MPS II, but it's really only helps patients without CNS involvement, which is actually the minority of patients with MPS II. Overall, there have been 9 enzyme replacement therapies approved. They're generally given intravenously. They generally require something between weekly and monthly infusions and cost upwards of $100,000 per year. That's for life, generally, for the treatment of these diseases. But I wanted to point out that specifically that in all of the syndromes in which the replacement therapy is possible, when it's administered intravenously, it generally has little or no impact on CNS or even peripheral nerve function. Next slide. I want to take just a couple of moments to talk a little bit about psychiatric diseases, because I've been talking about genetic, rare diseases, and neurodegenerative disease mostly. In psychiatric diseases, we've seen slower progress. One area that there's been a good bit of activity in is autism, driven by the genetic abnormalities of Fragile X syndrome. But the evidence for heritability in psychiatric disease is actually much stronger than it's always been in neurodegenerative disease in general, with schizophrenia, bipolar disorders clearly being inherited. But the problem has been that the GWAS and other studies in populations have generally been inconsistent. The loci are found, often just SNPs, and then they don't hold up in other populations and haven't generally led to targeted therapies. It's not clear why this is, but we don't have the same kind of neuropathology that we do in neurodegenerative diseases. So it could be that our syndromes are too broadly defined. depression may just be too many different drivers from a genetic background in order to pull it out of these kind of studies. The other possibility is that some of our diseases are too narrowly defined. One recent GWAS study had a much higher signal when more broadly patients were considered in relatives of schizophrenia who also had things like bipolar disease or other psychotic disorders. And of course, it may just be that the number of genes affecting behavior in the brain is too large, or maybe the impact of environment or resiliency is too large in psychiatric disease. And this may be a difficult thing for us to actually dive down to targeted therapies. Next slide. So we are seeing, based on this changing landscape, a change. First, final common pathway approach has yielded great drugs for many of the diseases in past years, but in general, we aren't seeing a lot of success with new targets using this approach. So companies, especially biotechs, but large pharma as well, are beginning to address these kind of small populations based on genetics. And we're seeing a variety of approaches, from more targeted CNS replacement of missing enzyme function, suppression of CAG repeats, including, for one company, allele-specific approaches, which is a subpopulation of the subpopulation in that disease, and then intrathecal RNAi, and small molecules as well, targeted for patients with a specific mutation, for example, modulators of GBA in patients who have GBA mutations. Next slide. But as I've alluded to, the challenges are combined here, the same kind of challenges that you see in rare and orphan diseases, of finding these patients, identifying them, enrolling in them, and then keeping them in what can be very small trials spread geographically. Genetic testing in diseases where these kind of risk factors are being developed has not been generally done because there isn't a lot of genetic counseling, and we don't really understand what risk goes along with each one of these mutations. And of course, many mutations are found in the same gene, so you generally need to sequence the gene and maybe coming up with new mutations all the time. And second of all, as I've alluded to, intervening in the CNS has always been a challenge for CNS drug deliveries. But now we're seeing with these targeted therapies some very novel approaches to delivering, demonstrating targeted engagement. And one of the things that we've learned from orphan disease and rare disease is that often when you target the actual cause of disease, the effect size in the trial is larger and smaller trials are possible. We don't know yet whether or not that's going to pan out here, but we can be hopeful. So now I'll turn it over to Michelle to talk about some of the operational issues that we face in these kind of trials.
Thanks, Dr. Vornoff. So when we're thinking about enrolling enough subjects into a rare disease study, it can seem like an insurmountable challenge, especially when you're looking at a subset of a rare disease, as Dr. Vornoff mentioned, or an ultra-rare disease population. By mapping out your path before you climb, you can mitigate a lot of potential issues. We've seen success in two key concepts in rare disease recruitment and retention. The first being making sure you're taking a patient-centric approach at the very beginning of the trial. So before you've even finalized your protocol, you need to understand what's important to your patients and how study burden can be reduced. You want to make sure that your design is appropriate with endpoints to make sure that your patients are going to be happy with the treatment that you're providing. The other key concept is referral networks, which you need to build them before you actually need them, because it does take some time to set up and maintain. You want to make sure that you're getting connected with the right people to make sure that there's a high awareness of your study, as well as you understand how you're going to allow patients access to clinical trials no matter where they're from. Because we're already working in such small groups, we need to make sure we're taking a global approach. So to address the concern of patient centricity, it's more than just a buzzword. We want to make sure that there's an appropriate risk-benefit profile from the very moment of your protocol design and in everything that you do. Some key benefits to participating in clinical trials can be highlighted by providing them extra care and extra attention, and making sure the risk-benefit profile is well understood by providing a well-written consent, as well as providing consent aids such as a video aid for what type of procedures will be done, or even illustrations of procedures, especially in the pediatric population, to kind of take away the fear factor for participating in studies. The next key item for being patient-centric is to truly make sure that you're reducing their burden. So primarily what we try and do is reduce time away from home, because that's time lost at work either for the caregiver and or the patient themselves. And if that's not possible, we want to make sure that we make travel as easy as we can by providing concierge-level service from door to door. We also can achieve reduced time away from home by having home nurse support and home delivery of study medication, if that's possible with the safety profile of your drug. We also make sure that we try and provide as much visit window flexibility as we can in the protocol to make sure that patients can accommodate the study within their schedule. We also encourage family engagement early on and make sure that we can accommodate the caregivers as much as possible. They may need to be provided with accommodations for their other dependents to allow for subject participation in the study if they can't travel on their own. The next key factor is patient engagement. So we do find that pulling together a group of patients to form a patient board, as well as their caregivers and the advocacy groups, really helps give you some great advice on smart protocol design and making sure that you're aware of key challenges so that you can have efficient operation of the study. We also recognize that the relationship with the PI/CRC is critical, so we really support the team in making sure that they have the time to dedicate to their patients and can answer and be readily available. We also do provide personalized support products that are designed for patients with their disorder, so that we're really trying to make the study easier on them and providing them with any sort of comfort that we can. Related to comfort, often in rare disease, patients feel isolated. You can really help them by providing them access to networking within their own groups, as well as counseling and even transport to advocacy meetings to make sure that they're connected within their community. When building referral networks, this is critical to begin at the start of the trial, as I indicated earlier. The way that we've done this in the past successfully is to utilize everything that we have available to us. So oftentimes there is registry data available, and this helps us understand who is treating your patient population and where. We also lean on the advocacy group's intelligence to make sure that we are addressing all the pockets of patients that they're aware of and their key physicians that they work with as well. We also recognize that our own investigators that we selected for our study have great ties within their own community. So we do ask them for feedback on if there's any additional investigators that we should be reaching out to for patients in this study. Medpace also has been working in rare disease for more than 20 years, so we do have a lot of experience we can draw from in a lot of different patient populations and can utilize our site list to help make the overall site list more robust. We also recognize that our sponsor partners typically are well versed in the disease before they enter it, and they know the community well. So they also share with us sites and doctors they've worked with, and we build that into our own referral network. We also leverage existing networks. So some rare diseases are fortunate enough to already have a network together, so we do work with them in order to get patients into our trial. Now, once you've built the network, you want to make sure that you actually get the patients out of that. So the way that we've been able to operationalize this is to train local staff on sponsor-approved messaging of the trial so that we get the word out about the study, and we regularly engage these investigators with in-person visits, phone calls, email updates, and newsletters. And we do make sure that we push these messages out at regular intervals so that the trial is at the top of mind at all times. And then for connecting caregivers, we want to make sure that the transferring physician is comfortable with their patient going to a different facility, especially if they're traveling a long distance. So we do set up meetings between the transferring physician with the new treating physician to discuss the patient disposition and any important social considerations. We have found that there has been a strong willingness to do this for free between the investigators because they really are looking to enhance their patients' treatment options. So that's been a very altruistic thing that we've observed that's been very positive. We also here at Medpace have developed a patient transfer process that includes a very expensive checklist to ensure all critical items are covered before we actually move the patient. We work closely with sites to make sure that things like visas are in place, if the patient has any special needs, that those are accommodated. And that really does help ensure that the patient has a good experience so we can actually retain them in the study. So I wanted to share with you a referral network case study and how that helped us enroll a phase 3 Duchenne muscular dystrophy subtype study. So you take a rare disease and you take an even smaller percentage of the population that we were targeting. And we did implement a lot of the key items that I noted in the building the network and getting results slide. So training the local associates to connect with that local staff really does make sure that cultural sensitivities are paid attention to and that personal relationship is built. And to maintain that personal outreach, the key thing here is really regularity. We want to make sure that they are updated in a regular fashion and the study is kept top of mind. And in this particular case, we were able to support cross-border travel for about 10 countries for patients traveling from one country to another to supplement our recruitment. These were all done successfully and really helped enroll our trial. We also made sure that the connecting and referring team step was done, and that took place in all cases and was really helpful in making sure that the process went smoothly. The other thing that I haven't talked about yet that Dr. Vornoff alluded to was genetic testing programs can be really helpful. And in this case, we utilized genetic testing program to help pre-identify patients that didn't already have the opportunity to be genetically tested, even though that's the standard of care. And then those patients were more likely to be aware of the trial and enter it once they became confirmed eligible. With this strategy, we did have a doubling of the recruitment rate at 2 different time points when we rolled out this strategy to reach out to our built-up referral network. So this is a small case study where we saw a huge impact from utilizing a referral network after your investigators have exhausted their patient population. Because what we commonly see in rare disease is that we have a physician that regularly treats these rare disease patients, and once they've exhausted their patient network, they run out of ways to enroll the study. So this referral network supplementation really does help drive the finishing of your enrollment. And so with all that being said, I did want to pass over the speaker to Dr. Shire on new approaches to CNS therapeutic delivery and monitoring.
Thank you. When developing a new therapy, we need to identify the patient, deliver the treatment, and confirm the treatment reached the target and had the desired effect. Molecularly defined rare diseases or subsets of more common disease require a strategy for selection of the individual patients with the appropriate biology. Large molecules or advanced therapies such as genes or cellular therapies may need invasive delivery platforms to reach their target, and delivery should not be taken for granted, but is often challenging to measure. We may skip this step if there's an easy way to assess drug activity. Unfortunately, for treatments intended to slow disease progression, clinical confirmation of efficacy may take months or even years, and confirming the delivery of our treatment and its impact on biological pathways assumes particular importance. How do we identify the patients? The diagnosis begins, of course, with the history and physical, supported by appropriate diagnostic procedures. Clinicians may be classified as lumpers or splitters. Lumpers believe it's best to group patients with similar presentations and avoid wasteful academic distinctions that may be based on minor differences in presentation, whereas splitters find meaning in these subtle distinctions. We've known for a long time that a treatment may work in only a subset of patients, but only recently have we begun to understand why, allowing us to move from trial and error to mechanism-based treatment selection and precision therapies. This molecular and genetic understanding may be shifting the balance in favor of the splitters. Disease is a consequence of inherited genetics and of the environment, driving the gene expression and the disease biology. Biopsy plays a role in understanding disease biology and selecting appropriate treatments in oncology and in some neuromuscular disease, but there are few opportunities to biopsy brain or spinal cord. Imaging, in particular molecular PET, can be useful for confirming diagnosis of diseases such as Alzheimer's or Parkinson's, but cannot interrogate a broad range of molecular targets. Even if we had ligands for all targets of interest, radiation exposure would limit us to a handful. So more generally useful has been the cerebrospinal fluid, which allows us to analyze a range of markers. Of course, we must always consider the impact on study conduct. When introducing CSF collection in a clinical trial, it's important to work with the sites to preempt any patient or family fear or resistance, or even investigator fear or resistance, to use appropriate needles to minimize headache, and to attend to factors that may impact the analyses, such as circadian fluctuation in various markers. And then beyond the CSF, the impact of environment may also be captured through analyses of circulating brain-derived microRNA and from protein and gene expression markers that may be found within brain-derived exosomes and microvesicles. These are approaches that may reduce the need for CSF in the future. Whenever we're considering orphan diseases or subtypes of common disease, key consideration is the genetics. And unlike many of our other biomarkers, germline DNA may be easily collected, and next-generation sequencing is allowing us to affordably cast a broad net, more completely exploring the genes of interest, some of which are captured here for Alzheimer's disease. And as Michelle indicated, these analyses may often be conducted in advance of the trial itself. Next. One of the main impediments to development of new CNS therapies, unfortunately, has been the lack of effective strategies for delivering through the blood-brain barrier. Next. The barrier constitutes a structural and physiological wall between the bloodstream and the CNS. It's a function of the perivascular processes of astrocytes, of pericytes, connective tissue cells, partially lining the capillaries and venules, and especially of the endothelial cells of brain capillaries. The capillaries of the brain possess tight junctions, limiting the flow of molecules from blood to brain. Delivery of small molecules is difficult, but it pales in comparison to the challenges faced by protein, antibody, gene, and cellular therapies. Next. If we can't get through the blood-brain barrier, can we bypass it by delivery of our therapy directly into the brain, or can we reach the central nervous system via the CSF? Both approaches have been tried with varying success. Neurosurgical delivery to the brain has included cellular therapies and a variety of gene therapies, typically administered using viral vectors. For example, prosavin, a lentivirus delivering 3 enzymatic genes to the putamen. These genes were selected to increase dopamine, the final common pathway, rather than modify the underlying disease biology. And while the approach appears safe, it's shown only modest efficacy. Treatments may also be delivered via the CSF, most commonly into the lumbar intrathecal space. This approach is routine for spinal anesthesia, and it's also been applied for advanced therapeutics. Nusinersen is an antisense oligonucleotide that modifies pre-messenger RNA splicing of the SMN2 gene and thus promotes increased production of full-length survival motor neuron protein in spinal muscular atrophy. This approach appears extremely promising for targets that are located near the ependymal surfaces, but there's limited diffusion to deeper structures. Next. There may also be opportunities to enhance the ability of our therapies to cross the blood-brain barrier itself, allowing for even intravenous administration. Lipid-soluble drugs may diffuse across the blood-brain barrier, and various molecular modifications can enhance the ability of small molecules to cross. Unfortunately, many of these same molecules are then subject to the effects of active transporter efflux. Carriers and transporters are also available that can help even macromolecules reach the brain. In particular, receptor-mediated and absorptive endocytosis may allow otherwise non-penetrant therapies to reach the brain. Next. Fusion proteins, including some of the enzyme therapies mentioned earlier by James, linked to these transporters are in development, allowing these large molecules to more readily enter the brain parenchyma, as are antibodies that have been modified to enter the brain without the massive systemic doses previously required for intravenous antibody administration. Another approach is CNS trophic viruses, able to reach specific targets and with specificity that can be further enhanced by modification of their promoter sequences to reduce off-target effects of what are still typically very large systemic doses. Such therapies hold the potential to modify the genome of the target cells, so instead of requiring a lifetime of administration, it's possible to administer just on one occasion. Next slide. We think we've delivered our therapy, but now we face the critical questions. Did it reach the CNS? Did it engage the target? Did it affect the biology? And most importantly, did it improve the outcome? Next. Did our treatment reach the CNS? It's possible to place microdialysis catheters within the brain to sample parenchymal extracellular concentrations, but this is highly invasive and therefore very rarely performed in patients. For selective therapies, PET labeling may confirm entry, and analysis of receptor binding may confirm brain entry and target engagement, and this approach has been commonly used for psychiatric therapies. It's common to measure the CSF concentration of our therapies, but the majority of CSF is produced by the choroid plexus, and drug concentrations do not necessarily reflect the concentrations within the brain. Next. So it's difficult to confirm brain entry, but what we really care about is a step removed, the impact on CNS biology. As it turns out, this is sometimes easier to measure. CSF, while coming largely from the choroid plexus, is formed in part from the brain extracellular fluid, and CSF markers of target engagement can help us assess the effects of our therapy. In this case, showing a decrease in CSF-A beta in response to a putative Alzheimer therapeutic. Lumbar punctures, of course, are required for the CSF collection, and while they're increasingly accepted, they're still invasive and, as importantly, do not permit frequent sampling. Plasma sampling for brain-derived microRNAs is an approach that's showing promise, as are brain-derived microvesicles and exosomes. These approaches allow us to non-invasively sample the cellular composition of brain cells just from a blood sample, allowing us to look serially at changes in gene expression and intracellular protein. And so we're really revolutionizing our ability to diagnose and our ability to track treatment effects. Next. And ultimately, did our treatment improve outcome? That's the bottom line. Indications with very few available patients need the best possible study designs. This is true for orphan diseases, and again true when subsets of common diseases become orphan. Clinical evaluation of the patient is critical. Accuracy and reproducibility can be enhanced by careful rater training and certification. Video exams may be useful as quality control or for central adjudication, and this approach is particularly useful if the intervention is difficult to blind, as in some of the neurosurgical treatments. Calibrated, quantified, Instrumentation enhances our ability to detect subtle effects. And then rating scales, in particular, patient-reported outcomes, are an important measure of the treatment's effect. And frequent assessment, facilitated by scales that have been accessed via personal computers or phones, increased precision. And bring your own device has become increasingly common in clinical trials. It alleviates some of the burden of trying to provide devices across large-scale trials and increases the patient acceptance of not having to carry multiple devices around. To supplement these reported outcomes, home monitoring, either using patient-worn accelerometry or a variety of other patient-worn sensors, or by using home video systems, allows us to have objective confirmation of the clinical impact. And of course, what we care about ultimately is to get the maximal useful data from our very small number of subjects while minimizing the burden on the site and on the subject. So talk now about how to pick up subtle signals. And that's very important in initial studies to guide treatment refinements, or when our disease is just going to be modifying the long-term disease course. But our ultimate goal, especially for invasive treatments, is transformative benefits. Next. And our resolve has been renewed by 2 articles in the New England Journal of Medicine earlier this month, reporting on the success of the intrathecal oligonucleotide nusinersen to slow the progression of spinal muscular atrophy, and of intravenous adeno-associated virus gene delivery to not only slow progression, but to dramatically improve the functional outcomes within subjects. Next. And with that, I thank you and turn things back over to James, who's been reading through your questions.
Okay, thank you very much, Rick. And so I'd encourage everyone to who's listening to submit questions if you have any. So we have a few, and we start out with a question for Michelle. So what would you say is the most common challenge in rare disease recruitment?
So we talked about already about how difficult it is to find patients after your investigators have gone through their own database. So the primary solution to that has been referral networks. But another challenge we've seen is if there's been no drug development in that rare disease space before, getting awareness for your study has been a big challenge. So you want to make sure you're connecting with both the physicians so you can access their database and the patients. So what we do is we first try and learn about the patient habits to find out how to best reach them. And that can be done by working with the advocacy groups or pulling together a patient board. For physician awareness, we found that gaining presence in the scientific meetings that they attend early on has been very successful in making sure that they're aware of you and your study.
Okay, and thank you. And let me actually ask Rick a related question that came in. So we know in clinical trials, screen failures are always a challenge. How do you manage screen failures when you're looking at very small populations that may be a fraction of the usual patients going trials?
So not only do screen failures make it difficult to find these very rare patients, but they're also extremely discouraging for our candidate subjects and for our site personnel. Pre-screening is one of the keys, and this begins by identifying patients with the appropriate phenotype, so not just with the appropriate diagnosis, but also with other appropriate clinical characteristics. And this is followed by genetic screens. Performing our trials at centers where such testing is standard of care really helps us allow identifying patients in advance of study launch. And we work with our sites to ensure that they have the availability of appropriate genetic screening and that this can be implemented even before the study has started. Also, working at centers that are conducting multiple studies can be a great benefit. Often we think of sites that have multiple studies at the same time as representing competition. But when these different studies are recruiting patients with different genotypes, which will be appropriate for different therapies, that's particularly rewarding because if they don't qualify for one study, they'll qualify for another.
So one man's screen failure is another man's enrollment.
Absolutely.
Good. So actually, an interesting question came in, which maybe I can answer. I'll just read the whole thing. If I understood correctly, the approach for targeting subgroups is based on combined genetic profiles, but also clinical outcomes. For example, Fragile X syndrome individuals with FMR1 mutation may have different degrees of cognitive impairment. It seems tricky how you establish a consistent threshold to define a subgroup. So I think looking at it overall, one of the bases that we're seeing is that patients' treatment is being matched up with their pathology, and they're being, and that's being defined based either on biomarkers or genetic propensity. But it's absolutely true that then in these rare disease populations, we run into the problem that patients, especially ones that are in long duration, are at multiple stages of their disease, and not every endpoint is suitable. For example, a timed walk is only useful for patients who can actually walk. A patient with muscular dystrophy who is now wheelchair bound is not able to meet that endpoint. And so there are a variety of approaches that are being taken where target symptoms or different scales for patients at different utilities. And again, I think it goes back to what our hope is that the effect sizes that we see are so large that we're no longer trying to simply aggregate statistical evidence that drugs work, but rather in relatively small groups, we're seeing large effects. I don't know, Rick or Michelle, you have any other comments on that?
No.
No.
Okay, and another actually related question is, are clinical endpoints changing based on this genetic-based approach to CNS diseases?
We're seeing our clinical endpoints evolve, not just on the basis of the genetic approach, but based, as you were saying, on what is most appropriate for a particular population. We're seeing refined assessments in Duchenne's for upper limb assessments as we're enrolling some older children and adolescents into these trials. We're seeing a better understanding of how to design appropriate rating scales so that they're more sensitive to change and so that they're actually measuring what's of greatest interest to the patients.
And I think the other change that we've seen is that biomarkers were looked on as a way to see whether the drug had activity or what it was doing, but generally were thought not to be very useful in drug development. And now what we're seeing, for example, in Alzheimer's disease, where regulatory agencies are recognizing that early on, the large signal may actually be in the pathogenic biomarker, and then there'll be secondarily or in concert with that a clinical benefit that goes along with the effect on the biomarker. So I think it's changing how we look at biomarkers and physiologic measures.
Yes, and we've even seen some conditional approvals that have been based only on the biomarker change. And this is something that's now been established, at least in the United States, for disorders of high unmet need.
Let me answer another question. So what's the difference between a genetic risk factor and a genetic cause of the disease? So I think that's an interesting question that, as I've thought about it, I think in the end is really one of semantics. When you have a common disease in the population and you look at it from the point of view as if you have that gene, you have a 2 or 6 or 10-fold higher likelihood of getting that disease, we think of it as a risk factor. But when you think about ApoE4 in Alzheimer's disease, we see a very clear dose of the gene effect and also longevity effects, so that if you're heterozygote, you have something of a 25% risk lifetime of having Alzheimer's disease. If you're homozygote, it goes up north of 50% and possibly even into the 80s or 90s if you live long enough, so that the disease will be expressed. So we tend to think of those as causes, but what I think the real shift is that whether it's risk factor or cause, we're selecting therapy based on what the genetics tell us about the etiology of the disease and matching treatment to the patient. So if there are, I don't think any other questions are coming in. Oh, one more question. So I guess related to the clinical endpoint question, there's also a difference between very young patients and adults with the same genetic disease. How do you put them together in a trial?
So I can help a little bit with this. We have seen trials where we need to include both young patients all the way through adults. And what we do is we make sure that we have endpoints that are set up that are appropriate for both groups of people. And sometimes we do have to separate out the young from the older, because it's not possible, for example, for a young subject under the age of 3 to lay in an MRI not sedated still for several hours like an adult can do. So we take a look at other endpoints to make sure that we can at least get an idea of what's going on with the patient, see if there's an effect of the drug. Anything you want to add?
Yes, so for a new therapy that's just entering patients, we're often very reluctant to enroll the young children. But ultimately, the biggest benefit for disease-modifying therapy is seeing the younger you can enroll the subject. And so we'll sometimes have studies that may start off enrolling an older population, and as experience accumulates, they may migrate down to that younger group. And so having those age-appropriate endpoints just becomes paramount.
Very good. So with that, we can conclude, and let me turn it back over to Vicki to close out the call.
Well, thank you very much for those answers. And unfortunately, we have reached the end of the Q&A portion for this webinar. However, if you do have any further questions, please direct them to the email address showing on the screen right now, and that is info@medpace.com. And thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. And a survey window will be popping up on your screen, and your participation is greatly appreciated as it will help us to improve our future webinars. Now, please join us in thanking our speakers, Dr. James Warnov, Michelle Peterson, and Dr. Richard Shire, and you can do so by clicking the link in the chat box. We hope you have found this conference informative. Have a great day, everyone.